und unsere nächste Slammerin, die hat ihren Bachelor in Biotechnologie in ihrem Heimatland in Albanien gemacht. Anschließend studierte sie 2 Jahre lang Biotechnologie und Biochemie in Belgien, hat dort ihren Master gemacht, kam dann vor einem Jahr nach Marburg. Hier promoviert sie am Psychologischen Institut der Medizinischen Fakultät. In ihrer Freizeit reist sie gern, sie arbeitet im Garten und sie lernt gerne fremde Sprachen. Der Titel ihres heutigen Vortrages ist Gene Therapy, Delivering New Instructions to the Eye's Genetic Software to See Better. Begrüßt mit einem herzlichen Applaus aus Marburg, Nermina Xhaferri.
Okay, hello everyone, I'm Nermina. This is my very first Science Slam, so I'm really hoping that it will be clear enough for you to understand and enjoy it. I decided to give a small talk in gene therapy as a way to deliver new instructions to the eye's genetic software in order to correct for genetic disorders. First of all, I've shown this very simple comparison here to show that our eyes work exactly like a camera. So every object, I've shown this pink flower in here, is reflecting light rays which are focused from the lens into a light sensor. Exactly like similar to this, also in our eyes we have the lens that focuses the light into a similar structure like the light sensor in the camera that is called the retina. Okay, the retina is composed of photoreceptors, which are light-sensitive cells. They can be rods or cones, and they get information. So they get information from the light and transmit these light signals to the brain, either be the color of the object or the shape of the object, and then the brain puts all these information together to create an image, and this is how we see. But how do these cells know what to do? Of course, everything is instructed into the genome of the cells. Every cell has its own DNA, and segments of the DNA are called genes. Every gene encodes or instructs for the production of a protein. For instance, I've shown here gene 1 is instructs the production of proteins that are sensitive to light, called opsins. Gene number 2, for instance, may produce proteins that transmit the light to the brain, that are involved in the pathway that transmit the light to the brain. And then gene number 3 may be responsible for producing proteins that help nourishing the cells and providing all the energy that is necessary for a cell to function normally. But what could go wrong? All cells go through this process of DNA replication, and the main player in this process is this enzyme here, which is DNA polymerase, and this is adding these DNA building blocks called nucleotides, which can be adenine, cytosines, guanine, etc. It's adding them in a specific order. Sometimes this enzyme can go like oopsie and introduce an error in here. So instead of one nucleotide is adding another one, and yeah, this is the reaction. And in a more fancy scientific way, scientific term, we call these mutations. There are different kinds of mutations. Some of them can have no impact at all in the cell. Some of them can have either a good impact, like improving the function of the protein that is produced from this gene, but we're not focusing on them today. I'm focusing on the negative side, so the mutation that leads to the loss of function in these genes. For instance, if the polymerase is introducing a mutation in this gene number 3, this gene number 3 will wake up like this grumpy cat and will be like, nope, whatever I'm supposed to do, I'm not doing it today, whatever. And this leads to, depending on which gene will be mutated, there can be different kind of eye retinopathies or disease of the retina. But for easiness, I will call them today just in the general term vision problems. So we end up with different vision problems. What can we do? This guy suggests to lock him up in Azkaban, but we are scientists, we don't do that. So we say, let's find a solution to this problem. For that, let's think like a scientist. First of all, what problem do we have? What is the problem? This guy. So the different types of vision problems that we have. What is the cause? Of course, to find a solution, we need to go to the source of the problem, and our problem is the mutation in this particular gene who is producing a mutated protein that can no longer carry out its function anymore. And to fix that, what do we need? We need, of course, the normal copy of the gene instead of the mutated one. And this process where we introduce a functional copy of the gene into the cells is what we, in order to, of course, correct for genetic disorders, that is the main goal. This is what we call a gene therapy. Okay, so what do we need to perform a gene therapy? Like if I want to do a gene therapy in house conditions? Now that's definitely a joke because we don't do that at home, please. Okay, so we will of course need a functional copy of the gene, which is this one here, healthy gene, and then we need a transporter or a carrier that will we will be sure that this gene will go inside the cell. For this, we usually use viral vectors. Virus. Okay, so we put this gene of interest, which I've shown in orange, inside the capsid of the virus, together with the genome of the virus, and then we perform a surgical procedure, where we inject viral loads into the retina. And then the virus will get into the cell. That's what viruses do, we know that. And then the cells will be able to produce this functional gene. Of course, there are some aspects to be considered. We don't want to be bad luck Brian, who goes to the doctor to cure his eye disease and get a viral infection and end up starting a pandemic and infecting the whole world. Of course, an important aspect to be considered here is that we need to remove all the genes that make this virus infectious. So we call those virulence factors, and we remove those so we have no longer infective virus. So the virus will not be able to cause any disease. The next thing is to suppress the immune system during the treatment, because our body has its defense mechanisms, of course, and whenever a bacterial virus will get in, it will try to fight back, but we don't want this virus to be killed, because unlike any other virus, the average virus is this one is a good guy, and we want him to be able to get into the cells and express our gene of interest. Of course, there is also the maximum size of DNA that can be inserted in the viral genome. If we need to consider if it's big enough, we need to choose this vector or that vector that can accommodate a larger size of a gene. And for my last slide, I've brought this example of Voretigene neparvovec, which is an example where gene therapy has been applied successfully. It was approved by FDA in 2017, based on what I shown during my presentation, the steps that I've shown, we have this mutation in RPE65 gene, that's the name of the gene. And then this mutation caused the patients to develop retinitis pigmentosa, and they had severe visual loss. So the doctors would interfere and would inject this adeno-associated virus, AAV, into the retina to deliver the functional copy of the gene. And the result is that the therapy, of course, for the patients has been shown to lead to improvement of their light sensitivity. And that's the last slide. Thank you for your attention. Of course, this is me trying to put a funny thing in there, but I would be happy to answer any question if time permits. Oh, yeah, I finished on time. That's nice. I think that was it. Thank you. Thank you so much.